So we start with the regulation of the gene expression. What is gene expression? A gene is expressed when transcription and translation take place and an amino acid chain or a polypeptide chain is achieved. Whenever the surrounding conditions change, there is a genetic mechanism to respond to it. That is, the genetic expression is regulated and the genes can be turned on or off depending on the need for the gene product. That is the specific polypeptide a gene produces. Such control systems can be inducible or repressible. In inducible kind of control system, presence of certain chemical can induce the gene to produce the gene product, whereas in repressible control system, presence of a chemical represses the gene, that is, it turns the gene off and no more gene product is produced. Lactose metabolism is an example of inducible kind of control system where the bacterial gene is induced to produce lactose digesting enzyme whenever lactose is present in the growth medium. Thus, when lactose is absent in the medium, no gene product or enzyme would be produced. Under repressible control systems, tryptophan synthesis is a well-known example. Whenever amino acid tryptophan is already present in the growth medium, it will repress the gene and no gene product, that is the amino acid tryptophan, would be produced. The biosynthesis of tryptophan will only take place when it is absent in the medium. Before moving further, let's see what an operon is. An operon is a genetic unit consisting of a cluster of related genes. These genes might be involved in a particular metabolic pathway. Together with these genes is present a promoter and an operator region that controls the transcription of these genes. Under regulation of gene expression, we will first study the lac operon. The lac operon consists of three structural genes: the lac Z gene, lac Y gene, and the lac A gene. Lac Z gene encodes for enzyme beta galactosidase. This enzyme helps in converting lactose into glucose and galactose. Then the lac Y gene, which encodes for enzyme permease. Permease facilitates the entry of lactose into the bacterial cell. Then the lac A gene, which codes for transacetylase. Transacetylase helps in removal of related metabolic byproducts. Joshua Lederberg established that these three closely linked genes are in order Z, Y, and A. These three genes are transcribed together as a single unit, resulting in a polycistronic mRNA. Here we have a diagrammatic representation of the lac operon together with a repressor gene I. It has a regulatory region consisting of a promoter and an operator. There are three structural genes, Z gene, Y gene, and A gene, that encode for galactosidase, permease, and transacetylase respectively. So, the lac operon is regulated by both positive and negative control systems. In the year 1960, Jacob and Monod suggested a lac I gene that produces a repressor molecule on the lac operon. In the background of this information, we shall discuss the negative control system first. Here we have the lac operon together with the repressor gene or the lac I gene that produces the repressor protein. The repressor protein has two sites, one for binding to the operator region and the other one where the lactose molecule binds. Then we have the RNA polymerase, the promoter region, the operator region, and the structural genes Z, Y, and A. When the lactose molecule is absent in the medium, the repressor protein is free to bind to the operator region, which blocks the RNA polymerase from traversing further, and the transcription is stopped. Thus, no enzymes are produced. But when the lactose molecule is present in the growth medium, it binds to the repressor protein, causing an allosteric change, which renders the repressor protein incapable of binding to the operator region. Thus, the RNA polymerase is free to cause transcription, and the three enzymes are produced from the structural genes. Because the transcription takes place only when the repressor fails to bind to the operator region, it is known as negative control system. Let's see the positive control of the lac operon. Now, a cell will always give priority or preference to glucose when compared with any other sugar or carbohydrate molecule. Why? Because it is energetically efficient, which is because any other molecule will have to be digested first to achieve glucose ultimately. So, if glucose is present in the medium, lactose metabolism enzymes will not be produced. This is achieved by the catabolite repression, which involves the CAP molecule or catabolite activating protein and the cAMP molecule, the cyclic adenosine monophosphate molecule. The association of these two molecules gives rise to CAP-cAMP complex. The presence or absence of this complex regulates the transcription of lac operon. The cAMP levels are in turn controlled by the glucose molecule involving adenyl cyclase. When glucose concentration is high, the cAMP levels will drop drastically, whereas when glucose levels are low, the cAMP levels increase rapidly. So, when glucose is absent, lac genes are expressed, and when glucose is present, the lac genes will not be expressed as it is energetically efficient. Let's understand this further with the help of a diagram. When the glucose is absent, the cAMP levels increase drastically. The cAMP associates with CAP or catabolite activating protein, forming a CAP-cAMP complex. This complex associates with the promoter region of the lac operon. Once the CAP-cAMP complex is bound to the promoter region, the RNA polymerase is allowed to associate with the promoter region of the lac operon. Transcription is initiated, and once the mRNA is formed, after translation, we have the enzymes for lactose metabolism. But if the glucose is present, the cAMP levels will drop rapidly, no CAP-cAMP complex would be formed, and the CAP molecule alone would not be able to bind to the promoter region. The RNA polymerase won't be allowed to associate with the promoter region of the lac operon. No transcription would be initiated, and thus no enzymes would be produced.